What's up?
What's up?
My name's Hunter.
Evan.
And this is another episode of Enzyme School Time. This week's enzyme,
Catalase.
Ooh.
Today, catalase is known as one of the most common enzymes in the scientific world. However, it wasn't until 1818 when Louis Jacques Pinard discovered hydrogen peroxide that catalase was even noticed.
In 1900, Oscar Lowe first named the enzyme catalase and found that it was in most plants and animals.
To help you better understand catalase and its function in our human bodies, we are going to go over its structure in detail.
Then we'll go over to the whiteboard and we'll walk you through the mechanism through which it breaks down hydrogen peroxide step by step. Catalase has four heme groups, like what you might find in hemoglobin or myoglobin. These heme groups bind to an oxygen atom from the hydrogen peroxide molecule and facilitate the decomposition of the hydrogen peroxide.
The histidine and asparagine residues that you see here also assist with the reaction by properly aligning the oxygen molecules.
In step one, we have a hydrogen peroxide molecule and one of the heme groups. As the peroxide molecule approaches the heme group, the histidine and asparagine residues add a proton into the specificity pocket, allowing the heme group to attack the oxygen of the hydrogen peroxide molecule. Leaving an oxygen atom double bonded to the heme group along with regular H2O. A second hydrogen peroxide molecule approaches the oxide heme group and reacts with the oxygen. The new transition step breaks apart, and we have the heme group starting back at the first step along with an O2 molecule and our H2O.
The large majority of animals use catalase in every organ of their body. However, it's been determined that catalase is in its highest concentration in the liver.
Catalase can also be found in large concentrations in most plants and fungi.
This brings us to the most exciting part of the episode, which is the lab experiments. And today, we have two experiments to set up. One that involves chicken liver and the other a common food, potatoes.
But before we begin, we of course have to don our safety equipment and follow proper lab procedures. Kids, don't try this at home.
To set up the potato experiment, we're going to use 3 potatoes. One, which is raw, kept at room temperature. Another, we put in the freezer for at least 30 minutes prior to the experiment.
And the last one we boiled for 5 minutes.
Here are the 3 potatoes we'll be using for the experiment. This one on the left will be the one that we keep at room temperature. This one was taken out of the freezer. As you can see, it's starting to defrost. And this last one we'll be using to boil. So the raw potato we'll place into the beaker, cover it in hydrogen peroxide, and observe the reaction. For this experiment, we'll be using 3% hydrogen peroxide.
Next, we'll use the frozen potato. Next, a piece of potato is boiled for 5 minutes. After boiling this piece of potato for 5 minutes, we'll see how it reacts with the hydrogen peroxide. Using oxygen produced as an indicator of the reaction's efficiency, we can see the potato kept at rawest temperature, its catalase performed the best with the most bubbles produced.
The potato that was frozen produced fewer bubbles than the room temperature potato. This is because the colder temperature slows catalase's ability to decompose hydrogen peroxide. And as you can see in the last potato, the one we boiled for 5 minutes, there was little to no bubbles produced. This is because the excessive temperature decomposed catalase itself, causing it to be inactive. In the second experiment, we'll be using chicken liver and hydrogen peroxide to explain the concept of how catalase decomposes hydrogen peroxide and brings the reaction to a biological timescale.
The rapid production of oxygen bubbles is evidence of the extremely high concentration of catalase in the liver. Hydrogen peroxide produces dangerous free radicals in the organisms and is a byproduct of most metabolic processes.
In order to prevent this damage to our cells and tissues in our bodies, catalase is necessary to quickly convert harmful hydrogen peroxide into harmless water and oxygen.
Thanks for tuning in. This concludes our segment on Catalase, and we'll see you next time here on Enzyme School Time.